about, let me just briefly review a few things here. Get this started, is genetic structure. Okay, so in characterizing the genetic structure of a population, I gave you that there's 3 different ways to describe it. And I'm giving you a hypothetical example here of where we are looking at a single gene locus, where there are 2 different alleles that interrelate with each other with incomplete dominance to produce 3 different phenotypes and 3 different genotypes, and just only 1 gene locus. And to describe the genetic structure here, we can use the phenotypic frequency, which is simply the percent of the population that has a particular genotype. And so we did those calculations. It's the number of individuals with one particular phenotype divided by the total sample size. A little bit more useful is the genotypic frequency. And this here, although the numbers turned out the same in my example here with incomplete dominance of the frequencies, the numbers are the same. However, the meaning is different, because here the genotypic frequency is the number of a particular number of individuals with a particular genotype that composes the population. Now, since we're dealing here with incomplete dominance, there is 1 genotype to 1 phenotype. And so, of course, they do correspond, the same numbers, but still phenotypic frequencies, genotypic frequencies are different. A little bit more important that we're going to use a lot is allele frequencies. And so here we're not looking at, we get our allele frequencies by looking at the phenotypes that we have here, but still this is a more specific meaning. It's out of all the total alleles. And so we have 200 individuals in this population or in this sample, and each of them having 2 alleles at this gene locus. Therefore, totally in this population, we have 400 alleles. 400 alleles, 2 different types. And so out of those 400 alleles, how many of them are the big R and what percent are big R prime, regardless of the genotype combinations? Okay, and so we're using some mathematical symbols here, P and Q. P is equal to the frequency of big R. Q is for the frequency of big R prime. And therefore, we went through the calculations. And I pointed out that here you just count, since we have genotypes that correspond to the phenotypes, for the phenotypes, we can directly count all of the alleles. And I gave you 2 different approaches here. Here, you're directly, what I've written here on the screen, you're directly counting how many big R's there are out of 400 alleles. But we can also divide this by 2, okay? So these 2's will cancel out, and you have a different approach that still gives you the exact same result. In other words, you can take the 32 red individuals plus the one half of the 96 individuals. So you have to put, because if we're dividing everything by 2, that drops out, but then this has to be 1/2. Okay, and it gives you the exact same thing. Okay, different approaches to the same problem, and same thing with Q, and of course P plus Q equals 1. Okay, this is the genotypic frequencies, and we'll see that they're very, very useful. Now I'm going to go to a different example here. The first example I gave for determining the genetic makeup of the population was based on incomplete dominance. What happens if we have complete dominance? Let's go with that. Complete dominance. So here we now, I'm still using a flower, but in this case, there's just 2 phenotypes. There is red and there is white. And here, since we have incomplete or complete dominance, red is dominant, white is recessive. Therefore, our symbols are different. Big R indicating that it is dominant, little r that it's recessive. There, if we go out here into our population, we have 2 different phenotypes of our flowers. They're red, they're white. And we count them, and we have 960 that are red and 400 that are white. And therefore, these add up. Our sample size that we've sampled is 1,000 individuals. All right, now we can go through and calculate the phenotypic frequencies, genotypic frequencies, and the allele frequencies, where the phenotypic frequencies, easy, okay, because we only have 2 phenotypes, and we just count them out of the total. So the frequency of red individuals in its population is simply 960 individuals divided by the sample size, 0.96 or 96%. And likewise, the frequency of white individuals is simply the number of white individuals divided by the sample size, 4% or 0.04. And of course, that adds up to unity, has to add up to 1. No problem there, but phenotypic frequencies are not, that really doesn't convey real good information about the genetic structure of the population that we're really wanting to get at. So now if we go to genotypic frequencies and allele frequencies, do we have a problem here? How would we go about doing genotypic frequencies?
Would we need a test cross?
Say what?
Would we need a test cross to find out if something's heterozygous or homozygous?
Homozygous with regard to what?
In regard to the like if it's dominant, the red ones.
Exactly. Our problem here is that these red individuals, the phenotype that we actually see, it's 2 different genotypes. It can be big R, or big R, little r. Homozygous dominant, or heterozygous. We can't tease them apart. Just by looking at the phenotype, we don't know what genotypes they are. And therefore, we have to do something else. All right. So we have to take a different approach. Once again, I'm going to repeat the data so you don't have to jump back to your previous slide. Our reds, certain portions of them are big R, the genotype big R. The other one is big R, little r, and we can't tell them based on the phenotype. So we can't count them. One that we can do, however, is the homozygous recessives. Because with their homozygous recessives, we know that all the white individuals have to be homozygous recessive. And so we can get those, all right? And so if we have this formula, P plus Q equals 1, okay? P stands for the allele frequency of the dominant allele. Q is the allele frequency of the recessive allele. We can get Q, or we can figure out what Q is. However, we need to do something. If we expand this into a binomial by squaring both sides of the equation, then we will end up with binomial of this form, okay? So we're squaring both sides of the equation. We have P squared plus 2PQ plus Q squared equals 1. All right. Now, in this, if we make a few assumptions, mathematical assumptions, and I'll get into some of these assumptions a little bit later, we make some assumptions, then under these conditions, each one of these 3 terms represents a genotypic frequency. So in this case, P squared is the genotypic frequency of big R, that particular genotype. 2PQ is the genotypic frequencies of the heterozygotes. And Q squared is the genotypic frequency of the homozygous recessives, right? Now, this requires a certain set of assumptions, and we'll get to those assumptions later. Right now, we're going to accept that those assumptions hold true, and that indeed P squared does represent the genotypic frequency of big R. 2PQ is the genotypic frequency of big R, little r, and Q squared is the genotypic frequency of the homozygous recessives. Still, from our data is phenotypes. The only frequency here that we can get to is Q squared. Directly from our data, we can get to Q squared, all right? Because what we know is that Q squared is the genotypic frequency of our homozygous recessives. It's not the allele frequency of little r, because there are little r alleles in the red. However, this represents Q squared. So our 40 divided by population size that we have, it gives us the frequency of the homozygous recessive genotype, okay, which in this case is 0.04. How to get to Q, what can we do to figure out what Q is?
Square root.
Take the square root. Okay, so Q is equal to the square root of 0.04, and that turns out to be 0.2. Okay, so now we've figured out the allele frequency for the recessive allele, but now we go back to our, how do we figure out P? Well, we go back to our original non-binomial equation. P plus Q has to equal 1. We have already figured out that Q is equal to 0.2, and therefore P has to equal 1 minus Q. And in this case, P is equal to 0.8. And therefore, we have figured out the allele frequencies. Now, once we get these allele frequencies, what we can do is we can go back. See, did I go? Yeah. So we can go back to our binomial and insert into it, figure out what P squared is. In other words, we use this formula. P squared is the allele frequency of big R, the homozygous dominance. Okay, that's P squared. Out of our data here, we have 960 that are red. So what proportion of those would be big R? We have our P squared, multiply it by, not by the number of red individuals, because this P squared represents the genotypic frequency out of the total number, out of the sample size. And therefore, we multiply it by the sample size. So we've already calculated that P is equal to 0.8. So we take 0.8 square times our 1,000, and it gives us 640. So out of our red individuals, in which we have 960 of them, 640 must be homozygous dominant. We can figure out the heterozygotes in 2 different ways, and probably it behooves you to do it both ways just to catch any sort of mathematical errors that you may have made. One is that you can take, now to figure out big R, little r, how many individuals happen to have that particular genotype, is to take 960 minus your 640, because what's left over. But you could also still use this 2p cubed. All right, so let's do that. We're going to use 2p cubed times sample size. And so we have the formula 2, p was equal to 0.8, q is equal to 0.2, and multiply it by the sample size. And that, when you work it out, turns out to be 320. If you take 640 plus 320, it gives us 960. Okay, so this is the approach that you should do. Now, on any future problems, and you will have a few of these future problems to have to work out using this particular formulas, is the approach. Now, the approach, if you have incomplete dominance, you can see every allele. You can locate every allele. You can just simply count them to come up with the allele frequencies. But if we have this situation where where we have complete dominance, then this is the steps you take. Locate the homozygous recessives and take the number of the homozygous recessives, divide by the sample size, and you have Q squared. Always have to go through the homozygous recessives, never through anything else. Homozygous recessives, number of those, divide by sample size, gives you Q squared. Take the square root of that, that gives you Q. Go back to the first formula, P plus Q equals 1, and that gives you, you can figure out what P is. Then go back to the binomial and take P squared times sample size to figure out how many individuals of the dominant phenotype happen to be homozygous dominant and so forth. And that is the approach you take. Now, on any problem, you need to be very careful about what I'm asking for. So read very carefully the question, because sometimes I may ask you what percentage of the population is homozygous dominant. And I didn't ask you for how many individuals. I asked you for percent. I'm asking you for the genotypic frequency. Other times I might ask you out of the total sample size, how many individuals are heterozygous? And there you need to give me the number of individuals. So please make a distinction here between the genotypic frequency versus the number of individuals, because I can ask both ways. Okay, now, as I said, this is based on knowing certain or following certain assumptions, which I'll get into here a little, in fact, right now. Okay. Now, what does this allele frequency have to do with evolution? I started out here talking about microevolution. Well, if we have those mathematical assumptions, and not just only mathematical assumptions, but certain assumptions about how this works, then if there's no evolutionary change, then our allele frequencies at this locus should not change from generation to generation. Now, this was something that was worked out in the 1920s by two different biological mathematicians named Hardy in the United States and Weinberg in Germany. And they arrived at this, this mathematical model that can come fair. I mean, it's a model, and it doesn't always correspond with actual nature, but is a good way in which we can look at what's happening in real populations and be able to try to understand what's going on. And so this is called the Hardy-Weinberg theorem. And in fact, this P plus Q equals 1, and it's binomial, P squared plus 2P cubed plus Q squared equals 1, is referred to as the Hardy-Weinberg formula. Okay, the Hardy-Weinberg formula. Now, if we went to 3 alleles, then that formula becomes more complicated. And I'm not, in this class, I'm not going to go to any gene locus that has 3, but then our formula would be P plus Q plus R, where each of these refer to, and they'd have to equal 1. And that would be for 3 different alleles. So P is for 1 allele, Q is the allele frequencies for the second, and R is for yet another allele. And when we did do the binomial expansion, then we're going to have P squared plus 2PQ plus 2PR plus Q squared, and so on. You see, it's I'm not going to work it all out, but it becomes a little bit difficult. So in this class, just to understand what's going on here, but sometimes we do have 3 alleles, and sometimes we do have 4 alleles, and so we have to deal with that. But in this class, we're going to stick just with 2. Okay, so the Hardy-Weinberg theorem is that if we make 5 assumptions about a population, 5 assumptions, then allele frequency and phenotypic frequency will not change from one generation to the next. Then the population is not evolving. But if one or more of these five assumptions is violated and not true, then it can lead to the allele frequencies and phenotypic frequencies changing from one generation to the next. Okay, and so that's microevolution. The definition here of microevolution is we have a change in the genetic makeup of the population from one generation to the next. If allele frequencies are changing or genotypic frequencies are changing, then the population is evolving. Okay, so here I have, they will remain constant generation to generation, or the genotypic, there's a second case here. the genotypic frequency will not be distributed as according to this formula, p squared plus 2p cubed plus q squared. So I'm giving here two different aspects in which we can say that the population is changing, evolving at this locus. 5 assumptions. If they're all true, then our genotypic frequencies will not change. If one or more of these assumptions is violated, then we will have a change in allele frequency from generation to generation. Second aspect here, even if allele frequencies do not change, because there's one particular assumption that if it's violated, the allele frequencies will not change, but once we know the allele frequencies and calculate what the genome frequencies should be, the actual genome frequencies do not match. The genotypic frequencies have changed, and we'll come to that case a little bit later. So either way, this is how we measure microevolution. So if one of these 5 assumptions is false, it can lead to a change in allele frequencies. No change in genetic traits if all 5 of these assumptions hold, there's no change, then we say that this population at that gene locus is in Hardy-Weinberg equilibrium. So being in Hardy-Weinberg equilibrium means no change, no evolution is occurring, no microevolution is occurring. Okay, obviously, when we try to look at all the genes in the organisms in the populations, it's very daunting. You can't do it because we have too many gene loci, too many genes. But still, this can be applied to a limited set of genes. We're going to be staying here with only one gene locus when we're looking at it. So if there's no change, no microevolution, this is in Hardy-Weinberg equilibrium. And so this particular model gives us a means by which we can measure the rate and changes of evolution at the microevolution. Okay, so we can define microevolution as a change in the genetic structure of populations. And we can measure that. So I'm going to go into these 5 assumptions. What do we say here? Now, first of all, let me give some examples here of how this can be used to measure whether or not the population is in Hardy-Weinberg equilibrium or is it undergoing evolutionary change. Let's say that we compare the same population at 2 different times, separated by several generations. So we come out here to our flower population and say this year, and we determine how many red and white flowers, and I'm going to give the same data that I gave here. So we've already calculated on a previous screen, we already calculated what those were, in which this particular population, it was P equals 0.8 and Q was equal 0.2, first generation. And so we wait several years, several generations, maybe 5 generations. So we come out 5 years later, same population, have to be the same population. And we collect flowers or count them. And this time we have 1,820 that are white and 180 that happen to be, red versus white. And so our sample size is 2,000. So how do we go about calculating the allele frequencies? You tell me. What do I do first?
First we find Q.
Okay, how do I do it?
So you could take 40 over 1,000 and then you'll get...
We already did that. So that was time 1, because we already determined up here that P is equal to 0.8 and Q is equal to 0.2. That's something I did in my first example. That was our time 1. Okay, so I'm talking about time 2. What do we do?
So we could do 180 over 2,000 and then square it.
Exactly. So we would have to go through that. So let's just, well, let's go ahead and do it. Looks like I didn't, I thought I did it. Okay. So if you have calculators, you guys figure it out for me. 180, 200.
0.09.
0.09. Okay, that's Q squared So we have to take the square root of that, and that is what?
0.3.
0.3. Okay, 0.3. So that means that P has to be 0.7, right? Because P plus P minus Q has to be, or 1 minus Q will give us P. So comparing the P and Qs from time 1 versus time 2, have they changed?
Yeah.
Yeah, so they are evolving. And in what direction? Okay, it looks like the dominant allele is decreasing and the recessive allele is increasing. And that is not accidental. For some reason, something's happening in this population that is leading to a decrease in the dominant allele and an increase in the recessive allele. Okay, so we know that this population, from that alone, we know that the population is evolving. Okay, so yes, it is evolving. And so out of, in population 2, then how many individuals can we expect to be big R, big R? How do we figure that?
P squared times 2000.
Okay, so we have 0.7 squared times 2000. And what is that?
980.
Okay, 980. Is that 980?
Yeah, 980.
Okay, all right, yeah, I was looking at the wrong things here. So 980, so we can expect that 980 out of our 1,820, that they happen to be homozygous dominant. And I'll leave it up to you to figure out the heterozygotes. Okay, what is the heterozygotes?
840.
And they should add up to 1820. Okay, so that's how you go about it. All right. Now we could also, and I don't have an example here, I'll come back a little bit later with another example. We can also tell whether something else is going on in the population if we have just only one time span. Okay, if we knew, for example, up here that P is equal to 0.8 and our Q is equal to 0.2, if we know that, okay, we don't have to figure it out, but there's molecular ways we take blood samples and we can figure it out, okay? But then if and therefore we should have had 40, according to this, if we're going with P squared plus 2PQ, the binomial, it would predict that we would have 40 that are white, and how many that are red from previous screen? If you look back, how many do we have?
960.
No, that are big R, sorry. How many of them are homozygous dominant?
Oh, from the first time, right?
Yeah, from time
40.
one.
640.
640. And the ones that are heterozygous?
320.
320. Okay, this is what P squared plus 2PQ plus R squared happens to predict. But what would it be, however, if we had, say, 60 and only 300 and more of, let me see here, am I doing this right? I brought that down by that. Let's make this right, and maybe 700. I don't know if those add up, but I didn't work this out in advance. I should have. But nonetheless, if we had different numbers and then what is predicted, even though we know that it should be 0.8, 0.2, but it doesn't fit exactly P squared plus 2PQ. In other words, here we have higher numbers of heterozygotes than predicted and lower numbers of higher numbers of homozygous, lower numbers of heterozygotes than what P squared plus 2PQ plus Q squared would predict then something's going on here. And we'll come back to that. I'll explain what that pattern shows. Okay, I said there's 5 assumptions that Hardy and Weinberg came up with. And so their prediction is that allele frequencies will not change if, 1, the population is infinitely large. Infinitely large. And isolated from other populations. So the membership of the population, of course, it changes vertically, but not horizontally. So there's no members that are leaving the population, migrating to another population, or other members from another population coming in. This is isolated. There is no movement of individuals, because movement of individuals between populations move alleles, okay, and it changes the numbers. So, and furthermore, there's no mutations. Mutations change the alleles. Back when they did this, back in the 1920s, this was before it was really appreciated that Mendelian genetics had a lot to do with natural selection. That was before the major synthesis. It was at a time when biologists didn't think that natural selection was a big deal. Many people didn't accept that. They thought that the way in which populations evolved was by vast mutations. Even though they didn't understand fully the nature of the mutations, they were hopeful monsters that would sort of come up suddenly. And we now know that mutations, they're important, but they're not so important here as this assumption, but I'm still going to include it here, right? Because this is the original. Furthermore, another assumption is that mating is random. In other words, there is no preference between a big R individual mating with a big R individual, or heterozygotes with heterozygotes, homozygotes with homozygotes. The mating pattern is perfectly random. Okay, now this is a very important mathematical assumption, because if we have preferences in mating, it can change mathematically what's going on here. Okay, and going into the math to prove that is beyond what I want to do in this class. And the 5th one is that there's no natural selection. Okay, if all 5 of these assumptions are true, then for our population, then what should happen from generation to generation is there should be no change. No change in the allele frequencies, no change in the genotypic frequencies. In other words, this population should be in Hardy-Weinberg equilibrium. So I'm going to go through each one of these assumptions, and we're going to look at what happens if they're violated. And in a real populations, it might not be just one of these assumptions that's violated at a particular moment. It may be a combination of them, but still we're going to analyze these based on just one trait or one assumption at a time, okay? And see what will happen, okay?
Professor, if you don't mind, I just wanted to clarify in the formula p squared plus 2pq plus q squared is would that be the genotype frequency of RR?
Genotype, yeah, that's right, of the dominant, homozygous dominant. It's a genotypic frequency. So really keep these allele frequencies P and Q separate from P squared and Q squared. Okay, they're not the same.
Because
okay.
P and Q would be allele
frequencies.
And then we could only use Q because that's only separate because all of the homozygous are RR, right?
That's right, because that's the homozygous recessives. We know what their genotype is. Okay, if we look at the red individuals, we can't tell because we're using phenotype to determine genotype, and we can't do that with the dominant phenotype because there's 2 different genotypes that contribute to that phenotype. So the only way we can approach this problem is by, first of all, getting the genotypic frequencies of the homozygous recessives. And from that, we can get the allele frequency for the recessive allele and then work backwards.
Thank you.
Yeah, that's correct. Okay, so large population size. There's a concept here, it's called the gene pool. The gene pool out of all the breeding individuals in this population. This is the totality of the alleles that are available for sexual reproduction. Okay, so we're dealing here with the sexually reproductive population. And so all of the alleles, all the sperm, all the eggs that are produced by the reproductive individuals in this population constitute, all the gametes constitute the gene pool. Okay, each of these gametes are carrying one allele for the character that we're talking about. We're looking at single gene locus, and therefore that sperm or that egg can potentially be passed on to the next generation. All right. Now, what happens here in small populations? So let's say that we have allele frequencies here among the gametes, our gene pool. Our gene pool is 50% big R, 50% little r. So in other words, P equals 0.5, Q equals 0.5, all right? And so in the gametes, all the totality of the gametes will have that same ratio. 50% of the sperm cells and egg cells carry big R, 50% of the sperm and eggs will carry little r. And of course, sperm and eggs can only come together to form the next population. And this is based on just probability of meeting together, all right? Follows the rules of probability, just like tossing a coin. And so 50%, 50%, right? Now, what happens in the small populations, I've gone through this illustration before. If I have a coin, okay, my tossing it, it can come up 50% of the time, it should come up heads, 50% of the time it comes up tails. And so if I toss this coin 10 times, it theoretically should be 5 heads and 5 tails, but that's a small sample size. Small sample size, which corresponds to what happens in a small population. So small sample size, there's random deviations from that. It really doesn't necessarily represent the true outcome or the true population division here. So if I toss the coin 8 times, it's very possible, or no, 10 times, it's very possible that I could have 8 heads and 2 tails and still have an honest coin, okay? Because we get these deviations with small sample size, but if we increase the number of samples, if I sit there with that coin and I'm tossing it 10,000 times, then the number of heads and tails come very close to 5,000 each. Larger the sample size, the closer to the real ratios that exist. So what can happen in a small population is that you have a small gene pool. And therefore, just due to chance deviations in the mating or in the sperm and eggs coming together, is that you can deviate from the real ratio. And this is called genetic drift. It's called genetic drift. So what happens here with a small population from generation to generation is that the allele frequencies can change just due to random meeting of sperm and eggs, because it's a small sample size. And sometimes it can go up, sometimes it can go down, sometimes it can stay the same. But from generation after generation after generation, we get this fluctuation of allele frequencies. And it's due to sampling of a small gene pool. So if we started out with 0.5, 0.5, then in the next generation, just because of random drift, P might go up just due to chance, 2.6. And Q will be 0.4. Now, when this generation reaches maturity and starts to reproduce sexually, we don't have a gene pool out of which the allele frequencies for this second generation are 0.5, 0.5. Now, 60% of the gametes are going to have the dominant allele, and 40% of the gametes will have the recessive allele. And due to genetic drift, random sampling of this small gene pool, the next generation, P might decrease just by randomness, might drop down to 0.4. And so what we get is this sort of from generation to generation fluctuation. As long as this population stays small, what can frequently happen is you can lose one or other of the alleles. And we say that it goes to fixation. Now, this is a computer-generated study that it's showing a graph here. Generations are on the x-axis. Allele frequency, and it doesn't matter which allele frequency we're talking about. It could be P. Usually, traditionally, we say it's P. And so a P could be anywhere from 0, it's not there, to 100%. It's the only allele, okay? And if our allele frequency, if we're looking at just only 2 different alleles here, if P happens to equal 0.8, then obviously Q is 0.2. So this is based on the allele frequency of just one of those alleles, because if you know the allele frequency of one allele, you know the allele frequency of the other. Now, if it's a real small sample size, and now this has been iterated in a population, I haven't counted all the lines here, but I think there's probably about 25. So this has been starting with P equals 0.5. So we have about 25 populations here that we started with P equals 0.5. And if the population size is 20, very small, you see that from generation to generation, P is going all over the place. And the pattern is unpredictable. Sometimes, like this red one, after 10 generations, P has gone to 100%. In other words, we've lost Q. The recessive allele has been lost. And therefore, at this particular gene locus, we only have the dominant allele. And once you lose one allele from the breeding population, it's gone, okay? It doesn't come back, all right? So this has been gone to fixation. But there's other iterations here, very same, okay? Starting at P equals 0.5, Q, or the population size is 20, it goes the opposite direction. And we have several iterations here in which it gets fixed where we've lost the dominant allele, and therefore Q has become fixed at 1.0. Now, if we increase the sample size to 200, and we're doing the same number of iterations here, all of them starting at P equals 0.5, we still get a lot of random fluctuation here, but you notice that the amplitude of this fluctuation has decreased. And then if we increase the population to 2,000, larger gene pool, then it comes even, we still get genetic drift, but it is really within narrow bounds. And so this is the effect of population size. The smaller it is, can lead to genetic drift. And we have to have and so the requirement of an infinitely large population size, you can see the trend here. As we are increasing the population size, then the amount and severity of the genetic drift is really decreasing. And so if we were to go, which I did not do with this, to a population that is, say, 10,000, then this will come very close to a straight line. Okay, the larger the population size, the less amount of genetic drift. If we look at it a different way, the smaller the population size, the more likely we can lose alleles. And frequently, this is a consequence of certain small populations. We can lose one allele. Okay, I'll give you some examples here. So with this genetic drift, there's 2 different categories of how it can come back about. Something called a bottleneck effect. So if we had a big population originally, and in that population, P equals 0.5, Q equals 0.5, and let's say some catastrophe happens that has nothing to do with the phenotype of the organisms, okay? So it might be that everyone can be equally affected. So we kill off most of the population, and we started with a large population, but we only have 10 survivors. Those 10 survivors are a random sample of our gene pool, okay? And the survivors now may not represent P equals 0.5 and Q equals 0.5 from the original population, because it's like taking 10 random individuals and letting them survive. And now our allele frequencies have changed. Furthermore, now this is a small population, and as they breed and produce generation after generation, we have genetic drift. All right, this is the bottleneck effect. I'll give you an example of where this happened. Okay, elephant seals in the Pacific Ocean. There's basically two different populations. There's a northern population and a southern population. Okay, off the coast of Chile and South America, there's elephant seals, and we can count this as one population because these, even if they break up into smaller groups, they all eventually will mix with each other. In the northern hemisphere, okay, off the coast of California, we have elephant seals out here. That population was severely hunted during the 1800s for oil, because they have this blubber and they made oil out of it. It was a big resource. So they were hunted to almost extinction. By 1890, there was only 20 individuals left. Now, these two populations don't mix because they don't cross the equator. The northern population stays long coast of California and further north, whereas in the southern populations way down around Chile. The southern population was not hunted like this. The northern population was. And so we only had 20 individuals. This is a bottleneck because originally this population was well over 1 million or so individuals. So what happened? Now, of course, today, and this is a old number, probably from 10 years ago, about the population was 30,000. It's probably much larger now because the population has been growing. But nonetheless, all of these northern elephant seals are descendants of only 20 individuals. They all came from just these 20 individuals. And as a consequence, there was probably a lot of genetic drift. So sometime back in I think it was probably the 1980s or so, probably a little bit earlier, there was studying the diversity in this particular population by looking at enzyme. So they looked at 24 enzyme loci that and to find out how many alleles are in each of these loci, and it was just one. So for each of these enzymes, all these elephant seals of their sample that they take, they didn't sample 30,000, but they took a large enough sample, blood samples, looking for these 24 different enzymes and finding out how many isozymes, how many different versions of this protein. And they only found one allele at each of these 24. Okay, now that could be accidental because we do have some genes that we only have one allele because changing the protein changes too much. It's selected against. But when you compare this to the southern, okay, yeah, I guess I don't have it up here. They also did this with the southern seals, okay, the southern population at the same 24 loci. And all 24 loci in the Southern Ocean had multiple alleles. They had at least two or sometimes more for each of these. So how can this be explained? The difference between these two populations for the very same gene loci. This went through, the northern population went through a bottleneck, after which there was severe genetic drift. And one of the major consequences of genetic drift in a real small population is one of the alleles simply by chance wins, okay? And it goes to fixation and you lose it. Okay, so this is one example of a bottleneck. I can give some other examples of bottlenecks. For example, cheetahs in Africa. We know that sometime in the past, they must have went through a severe bottleneck because compared to other cats, African cats, such as lions and so forth, their genetic diversity at many loci, they have reduced genetic variability. Very many of their gene loci happen to have only one particular allele, whereas comparing that to other cats in Africa, that same gene locus, they have multiple alleles. And the way this can be explained is sometime in the past, the cheetah population must have been really, really reduced and then undergone genetic drift. And there's many populations right now. This is a sort of a conservation problem. The black-footed ferret Okay, if you know what a ferret is, it's a weasel-like animal. It used to be very, very widespread in the Great Plains, okay, all the way from Saskatchewan down to Texas. Had millions of these animals. Now there's only one colony left in Wyoming that only has a few individuals, and we mostly have these flat-footed, black-footed ferrets. They are in captivity. We have about 360 of them. That's all that's left of this species that once was very widespread. And of these 360 Blackfoot ferrets they have in captivity, they're all descended from about 7 individuals. 7 individuals, and they really have reduced genetic variation. And they're trying to use these to eventually build up the population and release them into the wild to try to bring back this population, because it's just a centimeter from extinction. And the problem with this is that once we have the population reduced so far, it's lost that genetic variability. And so a lot of these ferrets have a lot of, they're born with crooked tails, they can't walk, you know, it's a bunch of malformed conditions because they're highly inbred, okay? So that's another example of bottleneck. And we're getting more and more of that type of stuff happening due to too many humans taking over everything. Another way in which this can happen, other than what is called the bottleneck effect, something called the founder effect. And this is where we have the foundation of a new colony by immigrants into a new territory. So let's say that we have a species that this is its geographical range, okay? This is where we find them. And why do they not grow out here? Or why do they not live out here? Why do we find this species only within this area? Why not out here? Any ideas?
They're trapped on the island.
It's what?
They're trapped on the island?
No, this is not an island. Okay, this is mainland. Okay, so we have continuous land through here. But why do we find them only here and not there?
Food supply? Or like they already developed their territory?
Well, here's good territory that they don't have any competition with other members of the same species. Why did they not live out here?
Maybe there's like water.
Well, this is land.
No, like water supply.
Okay,
There.
let's look at California. Okay, let's draw, if I can draw California, pretend that's California. Sort of looks like it. Okay, redwood trees. We find redwood trees, they grow in this area, close to the coast. By the time you get up to Oregon, they might a little bit across the border, but you don't have them growing there anymore. Why do we not find redwood trees growing out here or over here or even up in Oregon? Why are the range of redwood trees just in this area? Close to mountains?
Are they competing with other species?
Okay, let's go to a more general thing. There are conditions there where they're at that allow them to grow to adulthood and be able to reproduce. Okay, in other words, the environment is suitable for their existence. And likewise with any animal, its range, they have to be able to live there. They have to be able to have the resources to be able to survive, be able to compete, and be able to reproduce. And what happens here, the best favorable conditions for that's probably going pretty much here in the middle of their range. But as we get closer to the edge of the range, then those environmental conditions, resource conditions, and so forth, they change. So that isn't the case that the environment, the resources are equally divided all throughout their range. We can find them growing in their range or living in their range where they have the resources that they can utilize and they can reproduce there. Now, out here, conditions are too different. They don't have the resources. However, it isn't the case that these environmental boundaries are very strict. Here might be the limit, but we have little patches out here. that have the right conditions, but they're not contiguous. They're not joined to the main range. If some of these animals, if we're talking about an animal or a plant, happens to venture out here and try to live, it will die, okay, because of real harsh conditions, not enough resources. But if we happen to venture into here, into this zone, this isolated little patch that is suitable, has the right conditions to grow there, then we can establish a colony. Now, what the founder effect is, if in the main population here, if the allele frequency happened to be 0.5 for P and put Q equal 0.5, if that was the allele frequency in the main population, we're talking about, with the founder effect, a small number of individuals making it here. Now, if this happened to be an insect, for example, a pregnant female, okay, that had eggs, this could be just one individual, and it would establish a new colony. This is a small sample of the general gene pool, and therefore the founders come here may not reflect the allele frequencies of the general population from which they came. And therefore, we already have a change in allele frequencies here. And afterwards, since it's a small population, it leads to genetic drift. Now, I'll go back here to the redwood trees because it's very complicated. For the redwood trees being close to the coast, they're dependent on summer fog. That's where they get the water, because we have during the summers long periods of no rain. But for these particular trees, redwood trees, they require the water that comes from ocean fog. Along the coast, it gets where the redwood trees we frequently have fog. Further down to the south, it just gets too dry. Okay, they don't have enough summer fog, and therefore these redwood trees die out here. Inland during the summer, there's just no fog, and therefore not enough water supply for them to exist. And so the redwoods, there's a limit as to how far inland they can be. Yes, Alquazim.
So although that spot is not suitable for the redwood trees, is it possible that like with genetic drift, a certain type of redwood tree is able to be
theoretical possible. The thing is, when we're dealing with redwood trees, they have such a long lifespan. The genetic drift changing things real rapidly is something that has a shorter generation time. Because one of the problems with something with long generation times like redwood trees or long lifespan like redwood trees is that we're looking at thousands of years, you know, sometimes from one generation. Well, they have overlapping generations, but sometimes environmental conditions change faster than that.
Thank you.
Yeah. Okay. And what's happening up here in Oregon? In fact, if you go up the coast here, our redwood trees are getting bigger and bigger and healthier. And sometime over, in fact, they're looking very good. They're not scraggly like down here in the south. They're looking pretty darn good up here. But along this coastal gradient, conditions are changing for another tree that's a good competitor, namely Douglas fir. We have Douglas fir here too. They're mixed. Okay, but up in Oregon, up into Washington, Douglas fir outcompetes redwood, and therefore the big trees up there are Douglas fir. So the whole point here is that in order for any population or any species be living, they have to have the right resources by which they can deal with it. And this is a big thing with climate change, because some of the local areas, they're changing very rapidly. And if it's a plant, then they have to spread by their seeds moving to another area. Or even animals, they have to either, within that area, if it changes too much, they either have to adapt, and that's quicker to do if we're looking at short generation times, or they have to immigrate to a place where the conditions are livable, or they go extinct. And right now, you know, this happened at the end of the last ice age. The peak of the last ice age in North America was 24,000 years ago. And then the ice started melting. And our climates have changed quite a bit for that first 7,000 years or so. And so yes, in the eastern United States, you found conifer trees way down in Florida, Georgia, and every place. And as the ice melted, those trees migrated north, so that now the species that were there 24,000 years ago, you mostly found them in Georgia. They're now way up in Quebec, in Ontario. That's where they grow. But during that period of migration, due to climate changes that are happening there were no human obstacles. Humans have taken over so much of the landscape that this is going to be a problem, okay? Because a lot of our land has been turned into agricultural land and also to cities and suburban areas, and that prevents a lot of plants from being able to migrate. Okay, we can say similar stories there, but I'm getting off the topic here. Okay, founder, go back to that. So if we have some that escape to a little place, it's a founding population, and that founding population is small, and it may not represent the same allele frequency as that was in the original main population, and we get genetic drift. Give you some examples. There is an island off the coast of Africa, in the middle of the Atlantic. It's not real close to Africa, but it's out in the middle Atlantic. It's a volcanic island called Tristan da Cunha. And no one lived there. It was a desert island. However, European ships trying to make the journey from Spain or England out to South America frequently would stop there to pick up water and other supplies. So, but no one was living there. So in 1814, so very early, there was a ship, a British ship, that landed there, and 15 people on this ship, males and females, skipped off and decided they didn't want to go any further. And so they stayed there. And most of the modern population of Tristan da Cunha today are descendants of these 15 individuals. So these 15 individuals represent a founding colony, and their allele makeup is not the same as the England from which they came, okay? Because, for example, among these 15 individuals, one had a genetic disease called retinitis pigmentosa. This is the autosomal recessive. And so that one individual, it leads to blindness, okay? So this one individual had this genetic defect, and we know that that has to be homozygous recessive. So out of these 15 individuals, if we assume, which we really can't, that the other 14 happen to be homozygous dominant, even if we do that and calculate the allele frequency here for retinitis pigmentosa, it is much, much, much higher than how it's distributed in England, the population from where they came from. So, and I think probably in England, I looked this up once, I think it's probably like 1 in 2,000 people have retinitis pigmentosa in England. Don't quote me on that, but somewhere around there. Here we have, in the founding population, 1 out of 15. And therefore, we already have a different allele frequency. And so by 1960, there was a study that the population of the island was 240, and most of those individuals were descendants of these original 15. And therefore, among these, there was 4 of them had retinitis pigmentosa. In other words, they were homozygous recessive. 6 of them were heterozygous. And this allele frequency for this recessive allele is 6 times higher than what it was in the original population in England from which these colonists were derived. And so this is founder effect, genetic drift, and the founder effect. I'll give you another example, because there's actually a lot of examples for this, but I'll give just one other example, is among Native Americans with the ABO blood system, there is no type B allele. And so they only can be type A or type O or they can't even be AB. This is among the original Native Americans. This is usually explained by small founder populations. We don't know that for sure. It's still argued, where did Native Americans, how did they make it here? What size of their populations? Right now, the best idea is that Native Americans originated in an area called Beringia, which is now underwater. Okay, so this is the area in the Bering Sea between Russia and Alaska. 24,000 years ago, this was dry land. And so this is where they came from. But still, the interior of the United States was covered by, or Canada down into the United States was covered by glaciers. And so there was no real way that they could come across land. But now there's more and more evidence that they probably got here by boat. And they may have gotten here from Beringia in small groups, probably, as long ago as 20,000 years ago. This is a still an intensive area of study. But nonetheless, small groups, it's very possible that very few individuals in this Collins group had the B allele, or if they had it, then the small group genetic drift could have led to its being lost. And so this is the explanation that's given there. We don't know that for sure. Okay, so large population size. Small population size leads to genetic drift, which does change allele frequencies, but not in any adaptive way. It's just random, right? And frequently, if the population is small enough, this is a summary here, it can lead to a loss of an allele. Second assumption here is no migration, because if we have individuals moving from one population to another, we're moving alleles and contributing to either taking away from a gene pool or adding to a gene pool. And so this is called gene flow. Now, use these two words very carefully, okay? Because gene flow and migration are not the same thing. They don't mean the same thing. Gene flow is due to migration. Migration is the movement of individuals from one population to another. But gene flow is a change in the genetic structure of a population due to genes being brought in from migration. Okay, so they're closely related, but keep the terms separate. And I'm going to give you an example. Yes, Alquizin.
You mentioned on the last slide about the Native Americans and then colonists bringing the B-type allele. Would that be gene flow or no?
No, because these are founder. There was no one here before. So they were founders. They weren't contributing to.
Oh, so the colonists are the Native Americans coming to Americas.
That's right.
Oh, okay. Not like Spanish colonists or anything. Okay.
No. We're talking about the Native Americans, the ones here first.
Okay, thank you.
Now, with Europeans, Africans being brought into the New World, that is gene flow. Okay, that is gene flow because there was an existing population here. Okay, but with the Native Americans, as far as we know, there was no humans living in the Americas at that time. So that's colonists. Okay, yes, Vanny.
So like Native Americans were like the indigenous Americans here.
Exactly.
Okay.
They were the first Homo sapiens to ever set foot on this continent.
And the European immigrants brought the B-type blood.
Yeah, in fact, the European immigrants brought a lot of things, like diseases that and other things. And this is gene flow, because a lot of time, and this happens, there's a lot of history of two different human groups coming together, mixing. One of the myth here is like the neo-Nazis or the white nationalist and racist is European purity. You want to keep it pure, the racism baloney. The thing is, the first humans who made it, first Homo sapiens that made it into Europe about 40,000 years ago, they did not contribute to the genetics of modern Europeans. Okay, we can take DNA from these remains of these first humans that got there, and we can tell a lot. We've advanced enough that we can look at certain alleles. We know their eye color, their skin color, and so the first you see these pictures of Cro-Magnon. Okay, they were blonde-headed and long hair and white. These real first humans that made it to Europe had dark skin, brown eyes. Okay, and when the ice expanded and reached its peak about 24,000 years ago, these populations were forced out and they went extinct. In fact, modern Europeans don't seem to have some of the genetic diversity that came from that first group of humans that happened to come there. Then after 24,000 years ago, as the ice happened to go back, then mostly from the Middle East, some from southern in Iberian Peninsula where population had been pushed down. These were the ones who reinvaded. And then starting about 7,000 years ago, so these were hunter-gatherers that were there, and most of them had brown eyes and brown and darker skin. Then about 7,000 years ago, 10,000 years ago with agriculture in the Middle East, we had this wave of humans who migrated with agriculture from Anatolia, Turkey area, into Southern Europe and eventually mixed with a lot of these hunter-gatherers. And in fact, a lot of times they just simply took over. And so, and then about 3,000 or 4,000 years ago, there are some pastoralists, they're called the Yamana, that originated from somewhere in the steppes of Russia. They were pastoralists. They had cattle and horses. And this is a third wave of populations that then moved into Europe. And so the modern European population is a mixture of these secondary hunter-gatherers, the Middle Eastern farmers, and these pastoralists. And there's been other, we're all mixed together. And so what we have with the Europeans is something that's new. They didn't always exist. Light skin is very, very new. It came from a mutation that probably came up about 4,000 years ago and has spread, okay? There's more I can go on, but that's getting off the topic. Now, a little bit later on, I'll have more to say about human populations, but my whole point being here is that, and I want to emphasize this very, very strongly, is that all humans on Earth right now are closely, closely related to each other. And the idea that we have races is a social concept. It's not a biological one, okay? Not a biological one in the least. And most of the groups of people that we see on Earth right now didn't exist as they are now genetically 15, 20,000 years ago, because the population structure of all humans, they've mixed and we've changed. Okay, Alquazin.
There's like examples of small populations that have been isolated for thousands of years on like islands and that like we can't visit. Or is it possible that they have like more differences due to like genetic drift in their populations than other human populations across?
Yeah, but I don't know of any examples like that because most of the islands that are inhabited by humans, they have been colonized relatively recently.
Okay.
You know, they haven't been isolated for a long, long time. Now, there is an interesting case here in Southeast Asia, between Southeast Asia and New Guinea or Guinea and Australia. There's an island called Flores. And at the Ice Age peak 24,000 years ago, and even beyond that, water levels, of course, were in ice caps, and so the water levels were very low. And a lot of these things in Southeast Asia that were now islands were connected by land, including this island called Flores. And after the ice melted, of course, water levels went up and we have all this isolation. On Flores, they have found a human skeletons that is not Homo sapiens. Okay, and it may have disappeared as recently as maybe 15,000 years ago. But some of the fossils there on Flores Island, they date from about 40,000 or somewhere around there. No, 70,000. I think some of the names go back that far. 70,000 to about 15,000 years ago. What we have on this island, and in fact, I think even during the Ice Age, this Flores Island was an island. And and it's a small island. And so these forms of humans that were there, they probably did not derive from Homo sapiens. Homo sapiens originated in Africa. moved out of Africa and started colonizing the world about 60 to 80,000 years ago. And but this group on Flores is they're called hobbits, okay, because they're very small, okay, very small, miniature humans, all right, and they share some characteristics with skeletons of another species of human that I'll talk about a little bit later called Homo erectus. And so we believe right now that this group of humans, these miniature humans, and this is not uncommon for miniaturization to happen to animals that they've evolved a long time on a small island. That's very, very common, because there's some islands where they have elephants that they're about the size of a German Shepherd, okay, small elephants. So this miniaturization on this isolated island is very common, and this happened to probably some offshoot of Homo erectus. And so we'll come back and talk about them a little bit later on. They were probably isolated for a long time. Okay, now why smallness? Resources. Okay, if you evolve a smaller size, it's better because there's limited resources on a small island. Okay, and so either the population will have to get smaller so that the resources can be shared among members of the species, or if you stay large, you just simply go extinct. Okay, I'm getting off track here, but does that make sense?
Yes, thank you.
Okay, so go back here. I'll probably just only finish this and then we'll call it quits for today. So gene flow, it's when we're moving into the genes from one population to another that can change allele frequencies. And I'll just give a hypothetical example here. So we have two different populations. And so this particular Hardy-Weinberg assumption was that the population is isolated. The population is isolated so that there's no migration. Okay, I'm going to say here that we have population A that consists of 100 individuals. And so P is equal to 0.3, Q is equal to 0.5. We have a second population, B, which has a different allele frequency. P is equal to 0.9, Q is equal to 0.1. Now, what I'm going to do here is have a one-sided migration. One-sided, I'm going to move some individuals from population B into population A. And so we're not having reciprocal migration. So I'm going to move 50 migrants, okay, 50 individuals, so that now our population A after the migration consists of 150 individuals, right? And let's assume, even though this is a small size, I'm going to rule out any founder effect, no founder effect. So among these 50 individuals, P is equal to 0.9 and Q is equal to 0.1. All right, so after migration, we need to determine what the P is after migration. I'm going to call that P prime. And so since we have 100 individuals here, and so that means we're dealing with 200 alleles, 30% of which are big P. So how many big Ps contribute from population A? 200 alleles, 30% of them are big P, or P, or dominant allele. So how many alleles do we have?
60.
60. Of our migrants, we have 100 alleles, 90% are the dominant allele. So how many alleles?
90.
90. So we add those together, 60 plus 90, okay? 60 of the alleles already existing in population A. And in population B, we're bringing in 90. And we now have 150 individuals, each of which have 2 alleles. Therefore, the total number of alleles is 300. And when we work this out, P now is 0.5. It has increased due to gene flow. Allele frequencies changed. And if we do the same thing, same argument with Q, Q prime is after migration. So of our 200 alleles in population A, 70% of those happen to be the dominant allele or recessive allele, so 140. And of our migrants, of the 100 alleles that they're bringing in, 10% of them are the recessive allele. That's 10 out of the total, and that works out to 0.5 as well. So you see, just by migration, gene flow, we do change the genetic structure when we define the genetic structure as change in allele frequencies. And this is happening very much among humans. And as I said, humans, it's not really that important because we're all vastly closely related genetically, despite the fact that we make such a big deal of what are really very, very minor physical differences and so forth. It's mostly racism, it's mostly social. That's people do that, but it doesn't have a biological reality. But still, we get a lot of movement and then admixture as they will mate with each other, creating other families and so forth. This is another way in which allele frequency could change. And it happens among plants and it happens among animals, other animals as well. Okay, I guess I'll do just one more, because this will be easy. Another assumption of the Hardy-Weinberg equilibrium is that there's no mutations, because what happens here with mutations is that mutations can change one allele into another. Now, when Hardy and Weinberg did this in the beginning, they just thought that the dominant allele could mutate into the recessive allele, and we get back mutation, the recessive allele turning into the dominant allele. But they had no idea of what mutation rates are. Okay, we measure them at, this is just one value I got somewhere, I have others, but the point is, and I don't expect you to know this rate, this is a very low mutation rate. And therefore, if we only allowed allele A to mutate into allele little a, to change the allele frequency from 0.5 to 0.49, and this is leaving out the reverse mutation of little a going back to A, big A. If we just only have one way, then it would take about 200 generations to change by 0.101. And so that's a very minor contribution. So as far as being a major force in changing allele frequencies and throwing down things out of Hardy-Weinberg equilibrium, it's not significant. However, it is significant in making new alleles. So we might start out with big A, and we also have little a, but with a mutation, instead of mutating into little a, we might now have A prime, a different allele, still dominant. And little a might mutate and create little a1. And this might mutate again, little a2. Okay. So my point here, mutations are still important for evolution because they create new alleles. Hardy-Weinberg, they assumed that you only had big A, little a, but here we get new alleles. Now, obviously, with these mutations, this is going to be a very, very small allele frequency when this new allele pops up. And the likelihood of it being lost by genetic drift is very high. But it could also increase in frequency if this mutation happens in a small population, because just by chance, genetic drift, it can increase very rapidly in frequency in a very short period of time. But in a larger population, this mutation may not have any chance. It may disappear. But in small populations, it can increase. Or if it has a very strong advantage to survival and reproduction, it can be selected for. We'll come back to that later. Okay.
I have two quick questions about this.
Sure.
When it mutates into a new allele, can this new allele could be dominant, right?
Sometimes it can, sometimes not. Okay, it can go either way. We could also have the dominant allele result in a recessive allele. That can happen too.
So in that case, could a gene at a certain locus become transition from complete dominance to incomplete dominance by mutation, or no?
Okay, these mutations, they would be, so you're talking within an individual.
Yeah.
Okay, see, at certain of the cells, if you're talking about somatic cells, but not germ cells, because these mutations, as far as evolution is concerned, these would have to happen in germ cells, okay, to be of significance. Any mutation that would happen in the body, okay, in the somatic cell, and that just happened, okay, that's where cancer comes from. Okay, so let's say that you have a gene, let's just call it E, and if it mutates, so you have two copies in every cell, and let's say it's homozygous, but if one of these happens to, I'm going to call it E prime, this might be a loss of function. Okay, so now we only have one functional allele, and it's producing one functional protein. I'm going to say that this is a loss of function, and this is a DNA repair. A DNA repair, it codes for a DNA repair protein that fixes mutations. This is one of the types of genes that if we have two recessive or loss of function mutations in them, then it can lead to the DNA not being repaired. When accidents happen, leads to cancer. All cancers are incredibly genetically out of whack. So this is still protected, but then in the very same cell, and all the descendants of this cell, they divide, they will now be heterozygous. And one of those cells, we would have to have the other allele mutate as well for loss of function. And then you have cancer, okay? But the likelihood of two of these alleles mutating that way is very unlikely, but it does happen. It does happen. Now, if we started out with an individual that already had, they were heterozygous, they already had one mutated allele, now it only requires just one mutation and you have cancer. This is the type of situation that is where the cancer runs in families. Okay, that's where the cancer runs in families, because everyone is inheriting one bad allele to begin with, and therefore it takes only one mutation to knock it out and throw it into cancer. Whereas this other case where cancer rises, we call it sporadic. There's no history of cancer running in the family, but it seems to be isolated. It does happen. This would be a case where we had two alleles that had mutated into a loss of function thing, but that is in somatic cells. Here with germ cells, that's a different story.
Can I maybe ask in a different way? If I have a gamete and then one of the mutations creates an A prime that's dominant, and then when through reproduction the other parent has transfers the other gene, the same gene locus, but it's not A prime, but it's A, but it's still dominant, and now it has two dominant alleles in the offspring. Would that be incomplete dominance, or would it still have one that if both of them are functional in that case?
Yeah, if this is the case that you had A prime A, it could either be incomplete dominance, or it could be it could also be codominance.
Okay, thank you.
Yeah, because they both would be expressed. That's correct. Okay. All right. So okay, I'm going to put that up. This is where I'm going to stop. Okay, this is also going to be, write this down, slide 38. So the quiz, it will go from here, from the beginning, slide 1 to slide 38. And so this will be the material for lecture quiz on Monday. And then we'll continue this. All right, so this is where I'm stopping. Anyone need to see a particular slide? I got off topic several times. I didn't mean to do that, but I find the conversation interesting, okay? And I hope you did too. But now we need to take care of what we're going to do on Monday. Okay, so I need to have you divide up into, if you already